can. So it's a great pleasure to introduce our speaker today, Professor Sebastien Roch. He is a distinguished professor, the last distinguished achievement professor in the Department of Mathematics at the University of Wisconsin-Madison. So he is affiliated to all the analytic departments, the statistics, computer science, data science. He got his PhD from Berkeley with Professor Mossa, hero of mine, and he got various positions in at UCLA, at Microsoft. He got a career award, lots of paper award, best paper award. I can say that he's a fellow of the Institute of Mathematical Statistics, where he gave one of the keynote lectures. He is on the executive committees at NSF for the big data science grants and many other things. I have the pleasure to meet Sebastien when he gave a seminal paper at the Recon conference a long time ago. And since then I followed his trajectory because he seems to be a force of nature. And we're so privileged that he works in computational biology. So I am delighted to see him here. He is leading several workshops at ICERM and will be here for a few months. So without further ado, Sebastien.
Thank you. Thank you so much for the invitation and for the introduction. Before I tell you what this talk is about, let me jump in and give you the motivation. This is how I usually start my talks with this little story that I'm sure many of you have heard before. These are Darwin's finches here, and there's a dozen or so of these species. They live in the Galapagos Islands, and the way that they have evolved, I think the cartoon version of it is something like this, where there was originally one ancestral species, maybe it lived on this island over here, and then at some point some group of birds migrated to a neighboring island, got cut off reproductively from the original group, eventually kind of adapted to a different environment and so forth, and formed their own separate species. And then the process of repeated itself, and maybe another group of birds went to this other island, maybe this subgroup went to somewhere else, and so forth. And so all of these produce different species of these Darwin's finches, and a very natural way to represent this evolutionary history, or this series of speciation events, is with a tree like this, or a branching diagram, where at the root of the tree here there would be this common ancestor that they also descended from, and then each branching here corresponds to one of those speciation events, where the original species stays there, and then there's a new one maybe that arrives. And then this process repeats itself until you get the current diversity of species, which is at the top here, the leaves of that tree. And so reconstructing these trees is a big business in computational biology. There's a lot of applications. These are used throughout biology to study various things. I won't go into the applications. I'm going to be interested in how you reconstruct these trees. And people have been doing this for a very long time. There's a whole series of methods that you can use to build trees like this in different contexts. But there's still a lot of challenges. And part of it is because you have nowadays genomics scale data that is both a good thing, because you can learn more about the evolutionary history of these species, but it also means that you have to figure out how to process this data and how to infer interesting things about this. And so there's a lot of work being done in this. Søren mentioned that we have a whole ICERM program, and there's a lot of people here in the room. I have to be careful what I say. There's a lot of experts on what I'm going to talk about, and I'll mention some of their work along the way here. But we have a whole semester with a series of workshops if you're interested in learning more about this and the kind of mathematics and the kind of theoretical computer science in particular that comes up in this. Okay, so this is what I'm going to try to do in this talk. I'm going to tell you first a little bit about what are these complications that arise from this genomic scale data. This is going to be sort of part tutorial, part survey, part new results. I'll focus on one specific question that somebody could ask, a very natural question to ask about the issues that come up in this area of building trees from genome scale data. The question is here, but it's going to take me a while to explain what it is, so I'll come back to what this is. And then I'll tell you about some of how our theory can help, in some sense, guide or better understand methods for doing this kind of reconstruction. This will stop me along the way at any point. As I said, this is partly tutorial, so I'm going to start from the beginning and just going to tell you some basic things. So let me just start with the basic object that we're interested in here, which is trees. Now, these are not just any trees. I'm going to be mostly interested in what I'm going to call binary phylogenetic X trees. Okay, so these are going to be binary trees in the sense that all of the internal vertices after 3. And you can generalize things to higher degrees, but it just gets complicated, so I'm going to stick with the binary. Now, the more important thing, though, is that these are leaf-labeled trees. So there's a bijection between the leaves here and some set X, which you can think of as the names, the species names. Okay, so here 1 would be dog, 2 would be cat, 3 would be frog, and so forth. Okay, so I'm interested in reconstructing this sort of thing. How do these species relate to each other? Now, sometimes there will be a root. It's very natural to root the tree, which is where the process sort of began in the past, I showed you. But often the kind of statistical methods that people use cannot necessarily recover the root, so I'm also going to be working with unrooted trees, and hopefully it won't be too confusing. And then, of course, we care about these objects up to isomorphism. And so it doesn't matter how you draw it, as long as it's the same tree, but also that the labels match, right? It's important here that somehow the human is next to mouse, and then the next one is lizard, and so forth. They would care about. I put some definitions in here. I'm not actually going to prove anything, so it doesn't matter if you don't follow the precise definition. Now, trees like this, one of the things that they kind of naturally represent is how far things are in the tree of life. And so it's very natural to look at a related sort of metric concept, which is that suppose that I assign, so this is one of those X trees. This one has a label on one of the internal vertices, so just ignore that. Otherwise, it's just a binary phylogenetic tree. And so if you put labels on the edges, like positive weights on the edges, then you can get a corresponding sort of metric over the leaves or the labels by, if you take 2 labels, say B and C, because it's a tree, there's a unique path between them on that tree, and then if I sum up the weights, I get a distance, quote unquote, between them, and I can do this for every pair, and I get this kind of table. This is going to be called a distance matrix. And one of the key things, as I said, it's natural to think of this as representing how far apart things are in the tree, and particularly you could think of this as the number of years or the number of mutations that have accumulated along these edges. Now, the other thing that's important about this, though, is that this kind of, you can go in both directions. That is, if I give you a tree like this with weights, it's easy to construct a distance matrix. But if I give you a distance matrix that actually comes from one of those binary phylogenetic X trees with weights on it, there's a unique tree that corresponds to it, and you can recover it. And it's not very hard to do. If you haven't seen this, let me just show you very quickly how this works. And since we're here to build trees, this is going to play a role. If you have suppose I only have 4 species. Okay, so I have a tree on 4 leaves. It's a binary tree. And so there's really only one tree shape, right? It's got to be this particular tree. But then I care about the position of the labels on the leaves, right? So whether A is next to B, or A is next to C, or A is next to D on the cherry. And my goal here is just to figure out which one am I looking at if I'm given some distance matrix. I know that it came from one of those 3, which one that it came from. And it turns out that there's a very simple way to figure this out using what's called a 4-point sort of condition. I'm going to take the distance between AB plus CD minus AC and minus BD. And now, why does this work? I remember that the basic property of these tree metric is that they're additive along the paths. Right? So if I take plus this one, but then I subtract that one, it means that the contribution from this edge disappears. And the same thing happens here, the same thing happens there and there. And what I end up with is twice the length of this middle edge. Minus is in the negative sign. And so if I do the same thing over there, so here I get something negative. If I do this over there, I get something positive. You can just check this is what's going to happen. You get twice the length of that middle edge. And then if I do this over here, it turns out I get 0. Okay, so if I'm giving you a tree metric on 4 labels that comes from one of these 3, you can figure out which one it is, at least in principle. Now, how do you do this more generally? Right, this is if you have 4 species. If you have n species, there's different ways of doing this again, but one of them is just to break the tree up into 4 tuples of species at a time, reconstruct one of these quartet topologies for each of them, and then you got this puzzle. Right? So for each quartet, you have one of these, and then you have to put them together somehow. And I won't show you how to do this, but given the expertise of this audience, I'm pretty sure you can figure this out very quickly, which is a little combinatorial problem to put these together. Now, in practice, of course, what might happen is that they might not be consistent with a given tree, right? Because there might be some errors and so forth, and some people look at more kind of combinatorial optimization versions of this. There's a lot of literature, theoretical computer science literature on how to do these sorts of things. Okay. Now, where did I get the delta in the first place? And where did I get this tree metric? This is where the genetic data comes in, and it looks, again, at a very kind of high level, it looks something like this. So this is the kind of data set that you might be looking at, where you have on each row, you have a different species. And so there's human, chimp, gorilla, and so forth. And then what you see over here is the DNA sequence of a particular gene. It's a series of A, C, Gs, and Ts. All of these species are related to each other through some ancestor. The ancestor had some gene. Think of it as the insulin gene, something like that, right? It had a certain sequence. And now, through the speciation and so forth, all these species were created. And along the way, errors were introduced in the genetic sequence. Right? So some C became a G, some G became a T, and so forth. And now, this is what you get in terms of current sequences of these, or at least one representative individual from each of these species. And there's a lot more going on here in that that orange arrow, how you go from the DNA to actually a nice neat table like this. There's a lot of algorithms in bioinformatics. But let's just pretend like this is what happened, just these substitutions that have happened. And somehow the idea now is that the further apart things are in the tree, the more mutations have accumulated since their ancestors. And so if I look at 2 species that are close by, human and chimp, I might have, say, only 3 differences between their sequences, and you can count just by looking at the color. There's a change here, there's a change there, there's a change there. And if I look at 2 more distantly related, these are 2 kind of monkeys, I don't know what they are, but you see a lot more differences between them. So they're further apart in the tree. And so it's very natural to think of this as, well, I'm going to count the number of differences, and that's going to give me some kind of metric that I can use to reconstruct the tree. It's not quite that simple, however, because remember that it's important that what I want is really a tree metric. It's something that's sort of additive along the paths. And here, if you look at if you think about what happens over here, right, you could think of the number of mutations that happen to be kind of roughly proportional to the length of the path. But I don't actually get to see all these mutations. I only get to see the 2 endpoints. And so it could have gone from C to T to G to A and then back to C or something. Right? So there has to be some kind of transformation that I have to do to get a proper tree metric from which I can use the theory that says that I can get back the tree. And so I'm a probabilist, and so this is the part of the thing that I'm most interested in, which is this is where the stochastic processes are going to come in. And it's very natural to use one of these continuous time Markov chains on a tree to represent this substitution process of how the genetic data itself evolve on the tree. Okay, so just because I don't have enough room here, I'm just going to look at the first 3 species here. And so I've got gorilla, chimp, and human. And I'm going to imagine that their evolutionary history is this tree here. So this is how they were related to some common ancestor. And now it's very natural to assume the following thing. So suppose that I'm trying to just kind of model one column from this data. There was some ancestor that I don't observe, right? I only observe what's at the bottom. There was some ancestor that had some letter there, A, C. And then what I'm going to imagine is that along the way, and so the tree itself is completely deterministic, it's fixed. What's going to be random is the substitution, so the actual nucleotides. So I'm going to imagine that there's a continuous time Markov chain here that just jumps at random between different letters. So here I end up with an A over here. It's possible that it was a G at some point and then went to A and so forth. But there's some process here. And then I'm going to imagine that this just repeats itself independently on every edge. Okay, so once I have an A here, then the descendants down here is an independent process from the descending, the substitution along this edge, and so forth. There's going to be some parameters attached to each. So for example, I could have a time that corresponds to how long it took to go from that speciation to that speciation. And there's going to be some other parameters, in particular, there's going to be some kind of mutation rate. At what rate do you jump from one thing to another? Okay, so I'm going to use here the simplest model, which is called a Juke scanner model, where every time you have a mutation, it's uniformly random among all of the other possibilities. Okay, so it happens at a certain rate, and then you jump.
You just what are the implications of assuming that the tree is fixed?
Yeah.
I mean, am I supposed to imagine that there's some bigger potential tree that it lives in, or something?
That's right. So this is I'm only showing you part of this tree. In reality, I've got all these other species, and so this is just going to be some tree inside of a much bigger one.
No, what I'm asking is, you're assuming that the tree exists, but of course the tree is supposed to have emerged from the mutations. Can you?
This is the point that's a little bit tricky, right? So my modeling here is going to be that I'm assuming that there's some tree that exists that relates these species, and it's just fixed.
I hear
and
you.
I'm just modeling the mutations on top of that. The story is going to get a bit more complicated later on, and maybe that'll help you understand better how this relates to the actual evolutionary process. I'm going to do it on top of it, so let me go faster. Okay, so all of this was to relate, the number of mutations along the way here is going to be sort of linear in t times mu, but I want something that involves just looking at pairs of leaves. And this is where I'm going to use Markov chain theory to say, well, the probability that I have a jump from A to G along this edge is some formula that depends on T3 and mu3. And it's this thing. Okay, so this is sort of the expected number of mutation up here, but the probability of seeing a change is this sort of nonlinear transformation. And now that I have a model for how the DNA evolves, I can. Oh, and one last piece is that I'm going to assume that each of these columns is an IID sample from this process. So they all have the same tree, the same mutation parameters, and I just get independent samples for each of these columns.
Just to clarify the model a little bit more, you're like labeling these with the nucleotides, and then you're assuming that the only mutations are IID, like A to G, sitting the bottom left edge, or?
Yeah. So I'm not showing you all the mutations that are actually occurring here. This A at some point might have become a C and then a T and so forth. By the end of this process, it was a G.
Okay. That makes sense. But you're doing this you're doing this simultaneously for all of the...
That's right, and independently. Each of these lineages, because they don't interact with each other, are just independent substitution processes. So how do I turn this into an estimator of the metric that's underneath? I'm just going to invert the formula that I had on the previous page. Right? So here, if I'm looking at 2 species, I can observe how many mutations there are between them, or how many substitutions by the end of it, right? Just by the end of this process, how many differences there are. This is what this thing here is. So sigma here is just my sequence. I is a particular column, and X and Y are the names of these species. So I'm counting how many differences there are between them, and then I'm transforming this to get something that's the inverse of what was on the other page. And now, if I take k go to infinity by the law of large numbers, this thing is going to converge to the probability of having a difference between them. And this formula will give you a consistent, statistically consistent estimate of the distance between species X and Y, where the distance is the sort of the sum of the t mu's along the. And once I have that, then I can apply any sort of distance method, for instance, to reconstruct it.
But you say this is the inversion of the previous matrix, but in the previous matrix, it seemed to be like you would allow different rates of mutations, while once you invert, you kind of assume that all the rates are the same, all the mu's are the same.
That's good point. So I'm sweeping things under the rug. I do allow these to be different, and you can show that somehow what's going to end up happening is that the difference, the probability of a difference between these two, is going to end up involving the sum of these. So there's something to prove there. But then once you have that sum, then it's just really just a matter of inverting this one. Does that make sense? I'm only showing you what happens along here because it gives a simple formula.
Yeah.
That's easy to derive. Otherwise, you have to run the formula like this and playing again and do it again here, and you just end up with the sum of these teams.
But you still are dependent on the different mu's. I mean,
That's right. And so I'm not getting the total time. Is that what you mean? What I'm getting is the total sort of expected number of mutations in some sense. But it doesn't matter because as long as it's a tree metric that corresponds to this tree, that's what ultimately I want to get back my tree structure. I may not be able to figure out what T3 and T4 are. That's a different problem. People do work on that too. That's a different problem. Good. Okay. Now, finally, I get to my complication, which is if you have entire genomes or a large fraction of genomes of these species, now you have multiple genes. Right? I was thinking about one gene. Now I might have many genes, and it turns out that different genes will tell you different stories about the evolutionary history of the species. Okay, so from now on, I'm going to have two different kinds of trees. There's going to be the species tree, which is the tree that was on the first slide. Right? This is the actual series of speciation, the evolutionary history of my species of interest. I call that the species tree, and it's always going to be one of those fat trees. You'll see why in a second. And then I'm also going to have what I'm going to call gene trees, and these are there are going to be thin trees, and these are meant to represent the history of a particular gene, sort of lineages of a particular gene. And it's easier to see on an actual example. And maybe this is going back to your question a little bit about what's really going on in the background. Right? My fat tree here is this sort of sequence of speciation events that has led to these three species here, human, chimp, gorilla. But now, if I look inside of it, really what I have are these different populations of different generations. Right? So these blue dots here are different individuals. As I go back in time, I get different generations. And eventually they meet inside of ancestral populations. And now suppose that I look at a particular gene copy, right? I take the insulin gene in one individual here, one individual there, one individual there, and I can sort of trace back in time where it came from. This particular human got it from his mother, who got it from her father, and so forth. And so if you trace back in time, you get, and you do the same thing for an individual in chimp and gorilla, these lineages are eventually going to meet inside of ancestral populations. And they're going to create a tree. And eventually they're going to choose the same parent. You get what's called a coalescence. And that's another coalescence. And at the end, you get this rooted gene tree. And so this is what I mean by a gene tree, is the history of a particular gene. And it's important because that's really the thing that I observe. I don't observe the fat tree. The thing that I observe is the gene tree, because this is where the mutations are happening. The mutations are happening along this red line. And so when I look at genetic data, what I can reconstruct are these red trees. And now you can see that you can get conflicting information about the history of a species, by looking at this particular example here, like the true history that I would like to reconstruct is this rooted triplet here that shows that chimp is closer to human. But if I look at this particular history for that particular gene, just by chance, it turns out that these two lineages here did not actually coalesce inside the first ancestral population where they met. Instead, they coalesce much later back in time. It can happen. You can just see it over here. And what happened here is that the gorilla lineage met with the chimp first. And so if I now count the number of mutations that have occurred, this guy is going to look closer to this one than to this one on that particular gene. And so I'm creating a conflicting signal. This is not really the history of the species, the thing that I really want to reconstruct. Let's say you might be also interested in the gene trees themselves, but suppose that my goal is to reconstruct the species history, then I'm getting something that's conflicting, that's not consistent with what it really is. And once you have something like this, you can actually model and how likely it is to happen. I can have what's called the coalescent process, where I have inside of each of these populations, I have a certain number of lineages, and basically each pair meets at a certain rate independently of all the other pairs. So going back in time, you get a first coalescence, and then you get a second one, and so forth. So you can write down formulas for the probability of observing these. And if you look at those three genomes, you'll see that indeed there's a bunch of genes or a bunch of loci in the genome where chimp looks closer to gorilla than to human, and this is one potential reason for why that is. Okay, so now how do I reconstruct my species tree if what I have is the gene tree? The gene trees are easy to reconstruct. I told you one way of doing this, which was with these tree metrics. Now, how do I get the species tree out of it? Suppose that I have a collection of gene trees. So this is my coalescent process. Right, so this is showing you in a bigger example how you can create a distribution of gene trees inside of a fixed species history. And I'm also going to assume, and so what you do is you just do this coalescent inside of each population, and you stitch together the lineages, and you get a tree. Now, the other thing that happens is recombination. And so here, if I look at two different genes, certainly if they're on two different chromosomes, they will sort of make different choices. One is going to go towards the father, one is going to go towards the mother, and so forth, and they're going to get sort of independent lineages. The same is true for genes that are in the same chromosome if there is this crossing over that happens between them. And we're going to assume that the genes that we have in our data set are sort of far enough from each other that over time they become sort of independent. So their ancestral lineages are going to be independent. In other words, I'm getting an independent, identically distributed gene tree for each of my genes. Does that make sense? I'm looking at the insulin gene across these species, I get one tree. I'm looking at the hemoglobin gene, I can build a tree there. It's an independent tree, but from the same stochastic process. We're good? And so how do I do this? And so there are many methods for reconstructing the species tree from the gene tree. And this is one result that you can sort of a seminal result in this literature by people in this room who are at ICERM this semester. Suppose that you have, so this is written as a sort of an identifiability result. If you don't know what identifiability means, what it means it's a sort of standard statistics thing where it says if you have two different models, they're guaranteed to produce two different distributions. And therefore, if you have enough data, so now you can distinguish between them. And so the unrooted species tree topology, so this fat tree here, if I ignore the root, I can get back its graph structure with the leaf labels. Okay, so this is what I call the unrooted species tree topology, is identifiable from the distribution of unrooted gene tree topologies. So if I give you the distribution of this multi-species coalescent process, you can get back the actual fat tree behind it without the root. And you can turn this into an actual method of reconstruction by following the proof, which is that you do this, you go back to this idea of quartets, and you do a majority vote over quartets. So if I have, say, if I only have 4 species, again, these are the 3 possibilities for my gene tree topologies. And if I construct these across many, many genes, I can count, so suppose I have 10,000 genes here, I can count how often I get each of these. And the most likely one is the correct one. So if I have enough data, the correct one is the one that I see the most often in my data set. More generally, if you have more than 4 species, you can do the same kind of trick. So it turns out that this multi-species coalescence has this nice property that if you take a tree with n species, but you only look at 4 of them, what you observe is the same as what you would have observed doing the multi-species coalescence over 4 species. And therefore the same argument applies here. If I do this for every 4 tuples, I can apply this quartet majority thing and get back each quartet topology and then put them together into a full tree. And this is one of the more widely used methods in practice. There are variants of this called astral, where you sort of build these quartets and then you solve this sort of the problem of fitting them in the best possible way in the tree. The proof here says something about the anomaly. There's no anomaly zone for 4 species. I will come back to that in a second. Okay, so that gives me now a statistically consistent method. It's stated as an identifiability result, but if I do it this way, I get a statistically consistent method for given a sequence of gene trees. I am guaranteed to recover the correct species tree in the limit if I apply this procedure. Okay, so far what I've done is this, right? So I'm assuming that what's going on in the background is that there's this species tree that I don't know. It's something that's fixed. It exists. It happened. And now what I'm modeling here is that I'm going to have these different gene trees for all of my genes. These are my different genes that I have in my data. I've got some gene tree for each of them that comes from this multi-species coalescent process. And then on top of this, right, once I have a gene tree, what I actually observe is the genetic data, which comes from doing this substitution process, the juice scanner model that I mentioned before, over these gene trees separately. And the method, this is what I've just shown you so far, is just one way of reconstructing a species tree, which is to do this sort of 2-step approach, sometimes called a summary-based method, where I start from my genetic data, I construct gene trees for each gene, and then from there, I construct a species tree using that method. And now I can finally tell you what my question is do I lose anything from doing this? From going to gene trees to species trees, do I lose anything? Is that really the best way to do this? Do I really want to build gene trees, or do I not want to build gene trees and maybe go directly from the genetic data to the species tree? Okay, so maybe I'm losing information by doing this sort of 2-step approach. And the question becomes, well, as a theoretician, like how do you turn this into a theoretical question that I can give a rigorous answer to using the tools that I have? So good? Okay, so what are some examples of, so before telling you how I'm going to turn this question into a mathematical question, let me tell you what are some other ways of building species trees. And so I focused so far on these quartets and these tree metrics or distance methods. This is just one approach to do this, and then part of the reason I focus on these is because they're also very nice. I mean, they work well in practice, but they also they're very natural to work with as a theoretical thing to analyze. But more generally, you could do other things. And so, for example, once you have a model, so I'm going back to the gene tree problem. I'm giving you some genetic data. I want to build a tree out of this like this. Instead of doing this tree metric business, once I have a model for my data, I can just use a standard estimator like maximum likelihood estimator. So I can just look at the probability of observing my data, which is this product, because it's a bunch of independent things, of the probability of each of these, given a particular choice of tree and parameters, and then I can maximize over this likelihood, find the data, the tree and the parameters that maximize the probability of observing this data. That's also consistent. If I take an infinite number of columns, I'm guaranteed to recover the correct thing under some technical assumptions. Now, this particular method doesn't, maybe for computer scientists, it might not be like a good idea because it turns out to be an NP-hard problem. And now, instead of just doing this very simple quartet business, like I'm searching over the space of trees and parameters, trying to optimize this complicated non-convex optimization.
Well, for a computer scientist, I guess we would say the worst case version of this problem is anti-counterfeit, but all you see in nature are random instances anyway.
That's right. And so now you can ask, if I give you a data set that actually was generated like this, can I find the maximum likelihood estimator in polynomial time, for instance? And so the answer is yes, but in a contorted way. It's not a computational way of doing this. What I can do is to show that with enough data, this maximum likelihood estimator is going to be the correct tree, and I can build a correct tree using a different method. That's the sort of result that we have. Now, in practice, right, you don't necessarily have enough data. Your data might not fit the model, and so you want to be able to solve this problem reasonably efficiently. And so there's heuristics for doing this kind of tree search that actually work well and are very kind of scalable. And in practice, this is probably the most widely used method for building these gene trees, these heuristics for maximum. Okay, so how do I use this? So the way that it's actually often used in practice in this context, and I'm going back to the genomic context of having a bunch of genes, and I want to reconstruct that species history. What people do is they just ignore the problem, and they're just going to concatenate. And so you have a bunch of genes, you can just put them end to end into one big super gene, and then you run your maximum likelihood method. This is sort of what they do in practice. And I mean, of course, this is probably not going to work because it's ignoring the fact that these different genes have different. Right? So even when I say I'm applying maximum likelihood, I'm applying maximum likelihood as if it came from the same tree somehow. That there was just one gene tree that generated all these genes. That's not the case. So that's going to be an issue. Now, in reality, you might hope that this multi-species coalescent process is creating trees that are sufficiently similar to the original one in some way that if you concatenate and run maximum likelihood, you might get something that's pretty close to the truth. And I guess that's basically what they're hoping in practice. And it seems to actually work quite well in many circumstances. But in reality, as I said, these genes come from different trees, and you can prove that this particular method just doesn't work in the sense that it's not a consistent method. That you can construct trees where these gene trees are actually sufficiently different from the original thing that maximum likelihood will converge to the wrong thing. And this is where this anomaly zone comes in. What is the anomaly zone? It turns out that you can construct species trees where not only are the gene trees quite different from the original species tree, but you can construct it in such a way that the most likely gene tree is very different from the true species tree. So doing some kind of majority voting is just not going to work. It works if you have 4 species, but as soon as you have more than that, you start getting into trouble. And so, for example, if you have 6 species like this, and you have these very short branches, and these are very kind of natural scenarios, right? This is called adaptive radiation. You can have a series of quick speciation events, and you can get a situation like this. And now, if you think about this process, the fact that the branch is short means that there's a small probability that these 2 lineages are going to meet inside of it. It's not going to be a coalescence. And so they're all going to coalesce at the top here. And the top here is just going to run what's called the Kingman coalescence. It's just a standard coalescence in one population. And it likes certain trees more than others, and it's got nothing to do with the structure of the bottom. Okay, so so once you take that into account, it's actually quite hard to prove a result like this because you're analyzing maximum likelihood over the wrong likelihood. Then once you can construct an example like this and use some tricks, you get this kind of result. Okay, so this anomaly zone in particular was studied widely, including by people in this room, and sort of triggered a lot of work in this particular literature about the multi-species coalescence. Now, the other thing that you could do is to run maximum likelihood on the correct model. What does that mean? I have a model for how this genetic data was created, which is there's a species tree I don't know, it produces gene trees, and on top of that, I'm doing this substitution process. And once I can write down this model, I can run maximum likelihood or do some kind of Bayesian analysis over it, which means I have to look over the space of trees, now over the space of species trees now that fit best the data. And now that turns out to not scale very well. So if you try to do this, just computing this particular likelihood is very complicated compared to the other one was easier. And if you sort of start looking again over the space of trees, and this doesn't scale very well. So you cannot In practice, you cannot do this on many species or many genes. But it is one approach, right? So one question you might have is, in terms of how well this reconstruction is going to be done, is this method, even if it's hard computationally, is this method better statistically than the two-step approach? This is where I'm going with this. Now it turns out that this concatenation business is not completely hopeless. It is possible to construct other methods that reconstruct trees with theoretical guarantees that are essentially like concatenation, where you concatenate things and you construct. This is very closely related to the Duke scanner formula that I showed you before. And you can show that under some conditions, this thing actually gives you a tree metric, even though it shouldn't be, because I'm concatenating things and sort of, in some sense, ignoring the fact that there's different trees, but it turns out that works anyway. There's other methods, including methods that were developed by people here at ICERM. SVD quartets is another one that has a really neat kind of algebraic idea behind it, and also has the property that in some sense, it's sort of like a concatenation method that actually works. So my point is that there's different ways of building these kinds of trees using this data. Some of them come with guarantees, some of them are wrong. So there's one, okay, so this is finally my question. Does one lose anything by using the two-step approach? And as I said, I'm going to ignore the computational aspect of it. I'm going to think about the statistical accuracy. Now, measuring statistical accuracy is not a completely trivial thing here. If I run a certain method over a certain data set, I get the wrong tree. I could compute a distance to how different it is from the truth, but how do I turn this into something that I can actually prove something about? And particularly, I'd like to be able to have a measure that allows me to compare different methods, that this method is better at extracting the statistical signal than this one. And this is where, in the theoretical computer science literature in particular, a long time ago, people started thinking about this kind of question. Okay, so the sample complexity question. How much data do I need? How many genes, or how long my genes need to be guaranteed to recover the true tree with high probability? And in particular, how does that depend on the parameters of my original species tree? Okay, so this is an example of a tree that's very hard to reconstruct. It's the tree of birds, and there's a lot of controversy about various branchings in there. This is like 50 birds sampled across the whole tree of birds, and this is one estimate of it. And you can sort of see from this picture why this might be a hard tree to build. So in this case, they did the further analysis where they actually did things in terms of time. So it's not just mutations. What's at the bottom here is the time at which these speciation have occurred. And what you can see is that something happens like 65 million years ago. And suddenly the tree looks very different. What happened 65 million years ago? The dinosaurs went extinct. And suddenly the birds were able to kind of take advantage of all these sort of ecological niches, and there was this very, very quick diversification of birds. And you can see it with all these very, very quick branchings. And so quick branchings are hard because they create these short edges, and short edges have very few mutations on them, and therefore they leave very little signal in some sense. And the other thing that's hard is that it's happening deep inside the tree. And things that are deep are hard to reconstruct because, well, intuitively, I say you're losing signal as you go in this direction. And so what can we prove about this? So these are classical results. Laszlo is somewhere here, about this sample complexity business. Okay, so if I go back to my gene tree question, one can prove, this is not exactly how it was stated in this paper, but I'm going to do this here under the Duke scanner model. Reconstructing a phylogeny with high probability requires this many columns in my data set. So this is a constant, and this is scaling like 1 over f squared. What is f? Remember, here I'm going to be interested in the effect of short branches. Okay, so f is the length of this middle branch, the shortest branch in the tree. Okay, so in general, no method whatsoever can be guaranteed to reconstruct the tree with high probability as long if they don't have that much data. How do you prove something like this? In the high-level idea is you take two trees like this. Right? There's A, B. This one is AC with a short branch in the middle. And now what you're interested in is to look at the total variation distance between the distributions of the data over these two trees. The total variation distance, I didn't put I should have put the definition here. But it's basically finding the event that has the largest difference in probabilities between the two. And so this is somehow getting at the question of how accurate can a test be at distinguishing between these two things. This is a notion of distance between these distributions, and it's directly related to the statistical accuracy of any method. And now, if you give an upper bound on this, if you show that they're very close, then it tells you can convert this into a result about how much data you need in order to be able to reconstruct the hyperbola. And so here it turns out that you want to use what's called the Hellinger distance, which is related to the total variation distance, but has this nice sort of factorization property. Here I've got these independent columns. This allows me to go from k columns just to looking at one of them, and looking at one of them is just a calculation, and then I can just give an upper bound on this, and it gives me this kind of result. So this is an information theoretic result. It says any method will need that much data. And in particular, in this particular case, the methods that I showed you before, the distance-based methods and the maximum likelihood methods, are all going to match this particular thing. You can do the same sorts of thing for the question of building gene trees. Again, the short branches are what's going to create problems here. You can get upper and lower bounds on the amount of data that we need for different methods. And again, these can match. Now, what's really interesting about this particular setting here is that, and this is where I'm going to go back to this sort of proxy of how much data I need. How much data I need in this context, in this genomic context, actually depends on two things. Right? There's the number of genes, and there's the length of the genes, how many columns per data set. And so just as a theoretical analysis, let's just pretend like every gene has exactly the same length. Okay, so K is going to be the length of the genes, and M is the number of genes. And now I've got this sort of two-dimensional thing. I can ask where inside of this diagram it's possible to reconstruct, and which methods actually achieve that. So since I'm running out of time, let me go a little bit quick over this. You can do the same kind of information theoretic lower bound to show that certain regions cannot be achieved. And so as of a couple of years ago, this was sort of the picture that we had. One thing that we know is that the summary-based method that I mentioned earlier are up here. If I have 1 over f squared genes and each of them is this length 1 over f squared, these methods will definitely work. And so one way of formulating my question earlier, to build gene trees or not to build gene trees, is to think about, and when I say gene trees, I really mean gene tree topologies, is to ask the question, is it possible to do reconstruction below this? Are there methods that can actually reconstruct with less data in some sense, where less data could be some complicated combination of number of genes and gene trees? And it turns out that the answer is yes. So there is a region here where it's impossible to do things. Okay, so this is in log, and so it gives you this kind of weird relationship between M and K that involves this slope here. Let me not spend too much time on this because you're going to see what's going to happen in the next picture. But we have various methods that can sort of match, that can match these different regimes. So it's possible to do it here and there, and so forth. At least we have various theoretical results that point towards that. Now, that was, as I said, the picture as of 2022. Now, one natural question that arises, so we started doing this on the simplest possible models, and we tried to generalize as much as possible. And one question is, how complex do you need your model to be for your theory to be relevant to what happens in practice? So because you can make these models, and you're always ignoring various biological things, you can make these models as realistic as you want, and you can make them as complicated as you want. And what ends up happening is, of course, the more complex they are, the harder it is to analyze anything and say anything meaningful. So at some point, at what point do you have to stop? And so Max, who is somewhere in this room, looked at the question of making this model slightly more general. And this is by looking at what's called variable mutation rates across loci. And so each of my genes so far, I assume that they all kind of evolve at the same speed. But in reality, some genes evolve faster than others. And you can model this naturally by just scaling the gene tree by a random scaling. And so the process is going to be the same across everything, but some trees are just going to get scaled more than others. And you can go back to the same kind of thing. And what Max ended up proving is that this beautiful, nice kind of trade-off just completely disappears when we incorporate this particular phenomenon, which is certainly realistic and relevant to practical data. And so now we're back to sort of the drawing board of, if you want to include that particular aspect to your model, these gene tree topology methods are still up here. They still work in this context. They don't care about the fact that things are big. So some genes are evolving faster than others because they're just reconstructing the topology. That doesn't affect them. But now you can sort of ask questions about, well, what happens down here?
There's a lab. There's a lab of one by NDCS.
Oh, so we should.
Okay, let me finish then. And it turns out that you can do things here. SVD quartet in particular is a method that works with scalings. I don't know any sample complexity result that shows that it works at this 1 over f squared threshold. Another student of mine who's not here in her thesis developed a different kind of method that involves sort of data splitting sort of idea where you can do this. The kind of thing that you want, which is to look at the 4-point condition, which involves the sum of two distances. And by doing this sample splitting, you can somehow get things that involve a product, and you can go back to this 4-point method and get something. So we do have some answers of potential sort of estimators that might work in this whole. So since I'm out of time, let me stop here and just do a shameless plug. I have this new book that came out a few months ago that I think will be of interest to theoretical computer scientists about discrete probability, random graphs, and markup chains on graphs. Okay, thank you for that.
Questions?
Copy X, the room.
Maybe we have only one question before we leave.
I have some questions, but maybe we can have them out.
Yeah, some of them will be taken.
Yeah.
Thank you.